Stages of mitosis. Stage 1. Prophase. During the prophase stage, the long and thread-like chromatids during the interphase start to coil and become visible under a compound microscope as individual chromosomes. Each chromosome at this stage contains two identical sister chromatids, the result of DNA synthesis. The membrane of the nuclear membrane, dissolves at the spindle fibers are formed. Centrioles migrate at opposite poles of the cell. The nucleus disappears from view. Stage 2. Metaphase. During this stage, chromosomes move and align themselves at the center of the cell called metaphase plate. Spindle fiber connect each chromosome on its centromere to the centriole located at opposite poles. Stage 3. Anaphase. During this phase, the proteins that bind the two sister chromatids of each chromosome divide. This will be followed by the movement of the separated chromosomes toward opposite poles due to the shortening of the spindle fibers.
Stage 4. Telophase. During telophase, the chromatids, now called chromosomes, are located on opposite poles. Nuclear membrane forms around the chromosomes which start to uncoil. The spindle fibers dissolve and disappear. It is important to note at this point that each chromosome in the daughter cell is composed only of one chromatid.
Stage 5. Interphase. After the division of the nucleus, karyokinesis, the cytoplasm starts to divide, a process called cytokinesis. Cytokinesis in an animal cell and other cells that lack cell walls starts at the exterior of the cell moving inwards until the cell is divided into two cells. This process is called cleavage furrow formation. In plant cells and other cells with cell wall, cytokinesis starts at the middle of the cell and moves outward by a process called cell plate formation.
Stages of mitosis. Stage 1. Prophase. Stage 2. Metaphase. Stage 3. Anaphase. Stage 4. Telophase. Stage 5. Interphase. Stages of meiosis.
Meiosis 1. Stage 1. Prophase 1. Chromosome starts to coil and shorten. The nuclear envelope disintegrates. Homologous chromosomes pair by a process called synapsis. Exchange of genetic material crossing over may occur between homologous chromosomes when the chromatids are broken. Crossing over contributes to genetic variability.
Stage 2. Metaphase 1. The paired homologous chromosomes align at the metaphase plate. The chromosomes in pair are attached to spindle fibers.
Anaphase 1. Chromosomes break at centromeres and sister chromatids move to opposite ends of the cell. Telophase 1. At each pole during this stage, there is a complete haploid set of chromosomes, but each chromosome still has two sister chromatids. A cleavage furrow appears, and by the end of the stage, the parent cell has divided into two daughter cells. This separation of the cytoplasm is called cytokinesis. In some organisms, nuclear envelopes appear briefly at this point. This intermediate stage is called interkinesis, but in others, the daughter cells begin immediately to prepare for the second meiotic division.
Meiosis 2, Stage 5, PROPHASE 2. The nuclear membrane disintegrates. New spindle fibers are formed around the chromosomes.
Stage 6. Metaphase 2. The chromosomes align at the metaphase plate and are attached to their centromeres to the spindle fibers. Stage 7. Anaphase 2. Each chromosome is divided into two sister chromatids. The chromatids, now called chromosomes, move to opposite poles.
Stage 8, telophase 2. Nuclear membrane is formed around each set of chromosomes. Spindle fibers disintegrate. The cell undergoes cytokinesis.
When I to pray, I try to make the worst seem better.